Good morning. I'm excited to share our recent work where we develop a technique called Fourier synthesis to expand the speed and multiplexing capability of optical diffraction tomography pattern generation with the aim of realizing kilohertz rate volumetric imaging for a variety of applications, but particularly for studying the swimming of single-cellular organisms. Optical diffraction tomography is a quantitative phase imaging method for obtaining the 3D refractive index of typically biological samples. We place a sample in an imaging plane and we shine a coherent plane wave through that sample. Due to the sample's spatially varying refractive index, this writes a spatially varying phase shift on the beam, which we then read out using off-axis holography, collecting images like this. If we use a plane wave at a certain angle, we obtain one projected view through our sample. And by changing the plane wave angle, we can obtain a series of other views that we can computationally combine into a 3D refractive index. The individual views correspond with spherical caps in spatial frequency space. And so our computational algorithm can combine all of these images and must fill in any missing information that's missed by the imaging process. ODT is a technique that's matured over the past 20 years or so, and currently most state-of-the-art approaches rely on digital micromirror devices to generate the array of plane waves that are needed. DMDs have a variety of advantages, including highly repeatable pattern generation and pattern rates in excess of 10 kilohertz. However, the current pattern generation strategies using DMDs entail trade-offs between the fidelity of the patterns, the speed of the imaging, and the degree of multiplexing that the approach is capable of. Currently, DMDs are usually placed in a conjugate imaging plane to the sample, and patterns are generated by displaying a periodic diffraction grading on the DMD. This can either be done using a binary diffraction grading using Lie holograms, in which case the full speed of the DMD can be used, but the pixelated nature of these patterns generates unwanted diffraction orders, which compromises the quality of the patterns and the ultimate refractive index reconstruction. On the other hand, we can use grayscale patterns by time averaging several patterns together, which eliminates the unwanted diffraction orders, but entails a loss of speed, typically of 1 order of magnitude in the imaging. Finally, the fact that the DMD is placed in an imaging plane limits the number of plane waves that could be multiplexed in a single image, typically to fewer than 4, because using more complex or larger numbers of plane waves entails the creation of extremely complex diffractive grids on the DMD. Over the past year or so, we've been working on building an ODT instrument that uses an alternative pattern generation strategy to avoid making these trade-offs. So our strategy is able to combine extremely high-speed binary patterns with high-fidelity patterns, as well as multiplexing 10 to 100 plane waves in a single shot. A diagram of our optical setup is shown here. Instead of putting the DMD in a conjugate imaging plane, we put the DMD in a conjugate Fourier plane. And that means to generate a plane wave, we need to put a pattern that looks like the Fourier transform of the plane wave on the DMD. And that's a delta function or a small spot pattern. And that's exactly what we do. And due to the small size of this pattern, there's significant Fourier broadening, which eliminates the unwanted diffraction orders, typically present in binary patterns. And further, we can now change the angle of the plane wave by changing the position of this pattern on the face of the DMD. And so we can multiplex many plane waves together by combining many spot patterns at different spatial positions. Using this approach, we've obtained very nice results. Here I'm showing you a slice through a 3D refractive index reconstruction of a tetrahymena cell. Actually, there's a little bit more complexity to the patterns that we need to generate on the DMD, and that's related to the diffraction physics of the DMD. So a digital micromirror device consists of a 2D grid of roughly 1 million micromirrors, which can be in one of two states, either on, where they make a certain angle with the DMD face, or off, where they make a different angle. And these mirrors form a diffraction grating. So if we shine a beam of coherent light, that light is diffracted into multiple orders, and the strength of these orders is modulated by an envelope that's related to the angle of the DMD mirrors. Here I'm showing you an example for all of the DMD mirrors in the on state. If the DMD mirrors are in the off state, the diffraction orders come out at the same angles, but the position of the center of the envelope is different. This poses a problem for the small spot patterns that I discussed, because we're using only order 100 on mirrors and order 1 million off mirrors. And the fact that the diffraction orders overlap means that there's significant contamination from the off mirrors, even though their envelope center is far away from the desired diffraction order. And so to avoid this problem, we add an additional carrier frequency to the spot pattern, which you can see here. And this carrier frequency generates 2 additional diffraction orders for only the on mirrors, and then we select one of these mirrors, or excuse me, one of these diffraction orders using an aperture in our system. Using these carrier frequency patterns has several other advantages as well. It allows us to control the phase of this beam, as well as to change the angle by modifying the carrier frequency. And that will be vital to our multiplexing strategy. So using this approach, we're able to realize 2 distinct types of multiplexing. One is we can multiplex plane waves that have different positions in the imaging plane to expand our field of view. And we can do this by using several different spots on the DMD that have different carrier frequencies, as the angle or frequency of a pattern in the Fourier plane will map to the position in the imaging plane. On the other hand, we can do what I'll call angle multiplexing by putting a variety of spot patterns at different positions on the DMD phase, but using the same spatial frequency. And as I mentioned before, the spot position in the Fourier plane corresponds to the plane wave angle in the imaging plane, so we can produce a pattern of plane waves that looks like this. The angle multiplexing approach is very analogous to what's done in intensity diffraction tomography, where an LED array is placed in the far field and multiple plane waves are generated by turning on multiple LED arrays. We've tested these 2 approaches using our system, and we're able to produce nice position multiplexed images where we reconstructed several different fields of view. We should also be able to expand this multiplexing further to fill in some of the missing areas in this picture. And we've realized the angle multiplexing. Here I'm showing you an example where we've multiplexed 12 patterns, 12 plane waves in a single image, and get a reasonable reconstruction. Our computational reconstruction approach is as follows. We collect the series of off-axis holograms for different beam angles. Then we iteratively solve an inverse problem to reconstruct the 3D refractive index, or the scattering potential V, shown here. And we use an accelerated proximal gradient descent approach using the FISTA algorithm, where we're trying to minimize a cost function that consists of a datal penalty term as well as a regularization term, here using total variation regularization. And we encode the physics of the problem in the forward model, which enters in the data fidelity term. Typically, we either use a right-up approximation, which is linear and can easily be adapted to multiple plane waves in the sample, or a multi-slice beam propagation model, which is slower but can take into account multiple scattering and more easily handle complex phase fronts, such as in the position multiplexing case. We've profiled reconstruction quality versus degree of multiplexing. Here I'll show you an example where we keep the number of plane waves fixed at 24 using these different positions on the DMD face, but vary the number of pictures that we take and image 1 micron polystyrene microspheres on a cover slip. If we collect one plane wave per image, we get a nice reconstruction here shown in a single 2D slice. If we multiplex 3 plane waves in a single image, we get a qualitatively similar reconstruction, although there's a little bit of difference in the refractive index strength that we see. For higher degrees of multiplexing, we start to lose some resolution until we multiplex 12 plane waves each image and take 2 images, and we see a more significant degradation in the reconstruction quality. However, this isn't a fundamental feature of this multiplexing approach. It's a limitation in the iterative reconstruction strategy, and in particular in our choice for the initial refractive index guess. And so we're working on more sophisticated ways to initialize this approach, as well as considering some deep learning strategies which might be very effective for these highly multiplex patterns. As I mentioned, we'd like to use this instrument to study the high-speed single-cellular swimmers, looking at not only the swimming behavior, but trying to map out the flow fields that are generated in fluid when cells swim, using polystyrene microspheres as flow recorders. And so to that end, we've started to image diffusing polystyrene microspheres in solution. Here, showing you that we can nicely track these, we can image them at high density, and we can image them at volumetric imaging rates approaching a kilohertz, here shown at 650 volumes per second. We can see some nice physics here. For example, we're able to resolve how the diffusion coefficient changes versus height above the cover slip, which is indicating there are hydrodynamic interactions in the system. And most recently, we've included these tracer particles in solutions with E. coli, and we're working to extract the flow fields here. All of this work has been performed in Doug Shepard's group at Arizona State. Thanks to our collaborators, and thank you to you.